Department of Entomology and Wildlife Ecology at UD. Thank you for having me today. So I'm going to try to today quickly sum up some projects I've been working on. Whoa, okay, sorry, Microsoft Teams just popped something up in my window. Okay, anyway, on some work I'm doing on a disease called Chagas disease that's in 2 regions that are not considered to be endemic for the disease. And so since this is such a short talk, I'm going to just jump right in. If I can figure out how to there we go. So the first place I'm going to talk about Chagas disease is the island of Trinidad, and I'll explain where that is if anyone's unfamiliar. And then the second place I'm going to talk about is right here in Delaware. So first of all, what is Chagas disease? Many people are not familiar with it. Chagas disease is a vector-borne, is a disease that's caused by a vector-borne parasite. So by vector-borne, I mean an arthropod, or in this case, an insect, carries the parasite, and then this disease is unique in that it transmits the parasite through its feces as opposed to its bite. But a vector-borne disease then is, an insect that or an arachnid in case of a tick that transmits a pathogen, a human pathogen through some way, generally through its bite, in this case through its feces. And then once a person is infected with the parasite, and the parasite is called Trypanosoma cruzi, it's a protozoan. Once a person is infected with this parasite, it becomes, unless it's treated immediately, it becomes a chronic infection that our immune systems are pretty good at keeping it under wraps for a while, for many years. And then as people get older and the infection gets older, they tend to show their symptoms through either cardiac issues or gastrointestinal issues. And once it's a chronic infection, there's no way to treat it. It's just a lifelong infection. And for this reason, there's a lot of people infected in the world that nobody really knows, but the estimates center around 6 million people infected right now. So this map is the World Health Organization's map of Chagas disease cases in 2018. And what I want to highlight here is, so the disease is only transmitted vector-borne in the Americas. So all of these other dots you see in Australia and Europe, those are cases that immigrated there. But it is transmitted, it is endemic to all the Americas. But I want to highlight here that there is no data for any Caribbean island. And this is odd because there are vectors all throughout the Caribbean. But the official message of all the global public health agencies is that yes, there's Trypanosoma cruzi, which is the parasite. Yes, there are the vectors, but they only bite animals. They don't bite people. However, the issue with this is it's never been studied. This is an assumption. And then they say, well, but there's few clinical diagnoses. But then there's basically zero knowledge of the disease in the medical community. And so this leads us to this thing that I want to talk about today, the endemism catch-22, which is a disease is only considered to be quote unquote there if cases are diagnosed. But cases are only diagnosed when things need to happen that only happen for diseases that are there. So disease surveillance, public awareness, especially awareness among medical professionals to be able to make the diagnosis, then cases begin to be diagnosed. But that only happens generally after a disease is considered to be endemic. And so we're stuck in this catch-22. And this is something I'm very interested in. And so essentially, without reported and recorded cases, a disease isn't there. It's assumed not to be there. It's the old adage, you know, lack of evidence, evidence of absence of evidence is not evidence of absence. Knew I'd get there. So my work focuses in the Caribbean, focuses on the Republic of Trinidad and Tobago, which is quite a large, Trinidad is quite a large island. It's a dual island nation that's just north of Venezuela. Trinidad is interesting, especially from a geological perspective, because it is a chunk of land that broke off the continental shelf of South America, as opposed to being volcanic in origin. And so it shares a lot of the same flora and fauna as Venezuela and South America. And Chagas disease has been studied in Trinidad. I don't expect anybody to go through this slide in any detail with the time constraints we have this morning, but this slide is just meant to say that for over 50 years, T. cruzi has been found in humans, in bugs, on the island, but nothing has ever been really done about it from an official standpoint. And the last study of Chagas disease in Trinidad was in 1992. And so I asked, what's going on there now? Is it still there? Are the vectors still being found there? What is going on? And what needs to happen? So we did a pilot study where we collected the vectors of Chagas disease all throughout the island, and we tested them for the parasite. And then we also tested their intestinal contents to determine the species, the animal species that they were feeding on. So we found 55 vectors, and this was only in 2 weeks. It was a small study, but it was mostly this species called Panstrongylus geniculatus. And these vectors are interesting. In this picture below, that's my thumb. And you can see that it's sensing the heat of my thumb through this container. And so it has extended its proboscis, and it's trying to bite my thumb through the container. And so we found that 84% of these bugs were infected with the parasite that causes Chagas disease. And it's shown here, it's pretty fuzzy, but this is a Giemsa stained fecal matter with a really nice Trypanosoma cruzi that posed for me right by the ruler, which is really nice. So we found T. cruzi infected bugs at all of our positive collection sites. But the really striking result here was that we found that 57% of bugs were feeding on humans. And this is important because if you remember, the World Health Organization and all global public health agencies state that have a comment on this. They say that the parasite is only enzootic. It's only circulating among animals. But our results found otherwise. And so we found all of the bugs we found were in human homes. And so this, what is the current status of Chagas disease in Trinidad? We found Chagas vectors infected with T. cruzi in human homes with human blood meals. So we believe that people are likely being exposed to T. cruzi and merits more research and attention, which is ongoing and on the island. And we think it's time to rethink its official Chagas endemism status. And kind of bolstering this is right here, the Pan American Health Organization collects blood bank data for all of Latin America and the Caribbean. And so here and the blood banks test the blood for antibodies against Trypanosoma cruzi, the causative agent of Chagas. So here we have the blood bank prevalence in 2020 of all the countries in gray are countries that are considered by the World Health Organization to be endemic for Chagas disease. And at the bottom, we have Trinidad, which is its T. cruzi blood bank prevalence. and yet it's not considered endemic, and its prevalence is higher than 15 countries that are. And so we are right now working on building up to a human serosurvey, which is what really needs to happen in order to present evidence to these public health agencies to say, hey, look, this needs attention. But why do we care so much? Who cares if it gets this label? Well, we don't really care if it gets the label. What we want are the resources. So diagnosis, in particular, we need diagnostic and treatment resources. Blood bank detection is the only way to get tested in Trinidad. You can't go to a doctor and say, hey, can you test me for this disease? You have to go out of the country. Same with treatment. If you have Chagas, you have to leave the country, go to Venezuela or somewhere else to get treatment. The other thing is this endemic status, as I mentioned earlier, it raises awareness among medical professionals, and it also can kickstart community awareness campaigns. So if you find a bug, one of these vectors, you can say, you know what to do. And so now I'm just going to quickly, because I'm probably used up all my time at this point, I'm going to just quickly go on our work. We're doing similar work in Delaware because it's a similar situation, apart with the vectors, not with the people. So the United States is another quote unquote non-endemic Chagas country, but we do have 11 species of the vector that spreads Chagas, or the Chagas parasite, Trypanosoma cruzi. And these are all the states where it's found. And actually, it's also found in Wyoming and Iowa. This is not an updated map. This is the species that we have in Delaware. It's called Trichoma sanguisuga. And in 2023, I found the first Trichoma sanguisuga in Delaware infected with the Chagas disease parasite. And this wasn't like a great discovery on my part. I just looked for it. That's all I really had to do. So I was the first person to say, hey, maybe this is infected. And it was. So, and this bug was found, these two specimens were actually found in somebody's home. The one infected with T. cruzi was found under somebody's pillow in their bedroom. So that was a fright for them. And so that started my pilot study on this species in Delaware. And I'm really asking basic epidemiological questions. Where is it? Who's it feeding on? Where is it breeding? These are things we need to know to design vector control campaigns and vector awareness campaigns. We just need to know this basic information to know what to target. And so last summer, we went and collected bugs at a bunch of different sites where we suspected that they might be, and we found 55. We found them everywhere we looked. We got submissions from the community as well. I have flyers that I give out, and we extended the northernmost range of the bug by 14 miles with a community submission from Bear. And the T. cruzi prevalence in adults was, I think this is around 68%. But the interesting thing is, in the juvenile bugs, there was only 1 out of 36 infected. So it looks like the adults are really the ones that are carrying the parasite. And then we did find human blood meals in the bugs. 5 of the adults had human blood meals, suggesting that they are feeding on humans in Delaware. And all these 5 were also infected with Trypanosoma cruzi. So 8 had only T. cruzi. 3 bugs had only human blood, but they weren't infected. And 5, and they were all adults, they were infected with T. cruzi and they had human blood. So take-home message here is that T. cruzi infected triata means they are feeding on humans in Delaware. It's not that common. It's not going to cause the next pandemic, but it is something that we believe needs to be looked into further. And we are looking into it further. We've doing more field work this summer. And so this is just a summary of kind of the surveillance for Chagas that happens in the US. It happens in 6 states. This list shows 7 states, but it's because if you see this asterisk at the bottom, Massachusetts stopped their surveillance in 2014. And this is the number of autochthonous Chagas in the U.S. Autochthonous is just a fancy way to say vector-borne. In 1955 and 1998, there were 5 reported cases. And then from 2000 to 2020, there were 78. So what's happening? Are they increasing? I would say no, they're probably not increasing. I think it's just that, again, when we look for them, we find them. 74 of the 78 cases were from the 6 states that surveilled for the disease. So, it's the classic adage, if you look for it, you find it. If you don't, you don't. So, we are currently in the process. We have a bunch of data at the bioinformatics core right now. There, because we sequenced the intestinal contents from these bugs to know what animals they're feeding on, because that can give us an idea of where we can expect to find them. And we're building up to being able to develop some species occupancy modeling to be able to predict where we would find the bugs, where we would find infected bugs, and what risk factors might be. And with that, I will end. I just want to acknowledge all my collaborators, University of Georgia, Nicole Gottdenker, and then at the University of the West Indies in Trinidad, Rod Supal. In Colombia, I collaborate with researcher Omar Triana Chavez, and then at UD, I collaborate with a bunch of profs at UD, and then funding for this is supported by INBRE, an INBRE Pilot Award.